So this is the overall microbial community, biofilm, plaque, whatever you want to call it. We all have this, regardless of how scrupulous our oral hygiene is. And in most people, most of the time, this is a healthy situation. It's a mild inflammation that keeps this microbial challenge under control. And mildly inflamed gingiva is a healthy gingiva, a healthy gum. But in some people, some of the time, this is a direct precursor to this. And this is the destruction of the periodontal tissue, as you can see. Loss of gum, loss of the alveolar bone, which supports the tooth, which is really the hallmark of the disease. Eventual loss of the tooth, bleeding, pus formation, all sorts of unpleasant things going on. What is it that makes this healthy situation turn into this disease situation? Well, the classical kind of view, and we think this is still more or less correct. The classical view is that in health, in a healthy situation, you have beneficial bacteria. So you have a protective immune surveillance, like I say, keeps that microbial community under control. And so you have tissue homeostasis. But then things can change environmentally. There can be genetic predispositions, colonization with different organisms. So now we have pathogenic bacteria that are producing their own virulence factors. Now that leads to a non-protective immune response, big immune response, loss of tissue homeostasis, and destruction of the periodontal tissue. Like I say, we think that's more or less correct, but there's two major aspects relating to microbiology that this doesn't accommodate. And the first one is that the terms like commensal bacteria, pathogenic bacteria, beneficial bacteria, those are terms that we impose upon the bacteria. Those are not intrinsic properties of the bacteria themselves. The second part of this, that part of the microbiome that isn't really accommodated in this model is that the microbiome is not homogeneous. Okay, so the microbiome is not the same between different people. That's not so surprising, but it's not the same in the same person between different sites in the same body. And that's not the same even between disease sites in the same body. The microbiome is distinct. And it's also, the microbiome is not homogeneous structurally or architecturally. And so here's some fluorescent image analysis from Gary Borsi's lab at Forsyth with fluorescent DNA probes, just from a plaque from microbial community recovered from below the gum line subgingivally. And you can see there's all sorts of different organisms that are spatially constrained, that are distinct places in this structure that they want to be. Before I go on really to talk about what we're doing with regard to the properties of these structures, I want to just sort of kind of step back for a second and go through a couple of the premises that kind of underlie the basic research that goes on in my lab. So the first premise is that bacteria, like humans, like to be in groups, like we are, and they like to have specific partners. So they like to particular people that they're next to, and that's usually driven by physiological compatibility. They like to communicate with each other. And the communication can be physical, or it can be chemical, or it can be nutritional. And these interactions can be synergistic, whereby one organism is helping another, or they can be antagonistic, where one organism is being detrimental to another. And it's the sum total of those interactions that drives the overall properties of the community and the overall pathogenic potential of the community. And so these interactions among the organisms determine whether or not these mixed species communities, these heterotypic communities, have pathogenic potential or remain associated with health. It's hard to analyze the positions of these organisms in the different communities, and we can see which organisms like to be next to which other organisms. And we're particularly interested in the interaction between Porphyromonas, in particular Porphyromonas gingivalis, which is classically considered a pathogen, and some of the earlier colonizing streptococci, and in particular streptococci, which is classically considered a commensal. But again, those are terms that we use on the organisms. So I'll show you those are not necessarily accurate designations. But our other underlying premise is that all life exists in context. And I like to illustrate that this way. This is a picture from from a microbiology conference a couple of years ago. So we were just coming out of the COVID situation, but everybody, everybody had to find a place to sit. Okay, so everybody has a place. They have to be spatially, and they have to be a particular distance from other people. So spatially, this situation is very strictly constrained. Okay, and communication is very strictly constrained here, right? So one person's talking, everybody else is listening. Okay, but now you get a change in the situation. You get it, in this case, we would change in the nutritional substrates available, and now we still have spatial structure, right? Everybody's sitting in one particular place, but communication is very different, right? So instead of one person talking, everybody listening, now we've got everybody talking and nobody listening, more like. And then if we get into a different situation, right, sort of outside the confines of the conference or outside the confines of the gingival crevice, if you like, our structure is broken down, and communication's different. Some people, some species have a disproportionate influence on community activity than others, right? And I'm not going to call my good friend and colleague, Dr. Patempa, a keystone pathogen, but you can kind of see where I'm going with the, with the analogy. The situation is probably a little bit more like this. So we have a group of organisms, and the actual identities of the organisms are probably less important than the gene functions that they contribute. So we have a group of organisms that contribute to health or are present in a healthy situation. And these organisms are communicating with each other metabolically, physically. We can have colonization by some other organisms that unduly influence the activity or the overall properties of the community. And that eventually leads from a healthy situation, a healthy community, to a dysbiotic community, which leads to dysbiotic inflammatory responses and breakdown of the periodontal tissue. Like I said, we're particularly interested in a two-species model. Let's see if we can get this to play. Okay. Here we have in green is Porphyromonas gingivalis, and in red is the earlier colonizing Streptococcus gordoni. And P. gingivalis adheres very well to Strep gordoni, much better than to a lot of other species, and accumulates into these little architecturally distinct communities. So our first question was, does this have any biological relevance? Is this important? So we went to a murine model of periodontal disease. It isn't a brilliant model, but it's one of the few models that we have where we look at loss of the alveolar bone, right? So the alveolar bone, which is supporting the tooth. So this is alveolar bone loss. Infection, not much going on. Infection with Strep gordoni on its own. Well, Strep gordoni on its own is classically considered a commensal, right? So no statistically significant bone loss. Infection with P. gingivalis on its own. P. gingivalis classically considered a pathogen, and we do see bone loss. We see statistically significant bone loss, but really, it's not a whole lot to be excited about. It's a very variable model. Different strains of P. gingivalis show different effects. Different labs find different levels of bone loss. Our lab finds different bone loss. When we do it with different people, so you can see more bone loss in this, but it's still, do you really want to build your career on this? But when the exciting things for us was when we infected first with Strep gordoni, then with P. gingivalis. Now we start to see levels of bone loss, which are potentially biologically relevant. So we feel that in this context, Strep gordoni is really misclassified as a strict commensal. And Strep gordoni is more like an accessory pathogen, an organism that aids and abets, if you like, the pathogenicity of another organism. At this point, it became kind of clear what we needed to do. So we needed to look at P. gingivalis in this context with Strep gordoni, and we needed to look at P. gingivalis on its own, and we could compare the two, and then we would see what makes these things more pathogenic than P. gingivalis. I know to be not that straightforward, but first thing we did was an RNA-seq analysis of P. gingivalis, either P. gingivalis on its own or P. gingivalis in a physical association with Strep gordoni. principal component analysis. And there were some good things and bad things about this experiment. So the good thing was that clearly P. gingivalis with Strep rhodoni, at these points, was distinct from P. gingivalis on its own. Okay, so P. gingivalis is a very different beast when it's in a physical association with Strep rhodoni compared to when it's just on a laboratory medium. The second interesting thing was that they were clearly distinct over time. So things were happening over time that reflected an ongoing communication interaction between the two organisms. So that all was good. But then when we looked at the genes that were actually accounting for this variance, when we looked at the genes that were different in the two situations, it was kind of, it was kind of disappointing to be honest, because what we saw were genes that we already knew about, genes that were encoded functions that we already knew were important for P. gingivalis.
Between P. gingivalis alone compared to P. and
Yes.
Today, there is an increase of bone loss.
Right.
One alone,
yes.
That into any bone.
Right.
You must have a pathway by increasing bone.
Right.
P. gingivalis alone and P. and
Yes.
Did you find that?
Yeah, I mean, there certainly were genes in common, but there was nothing that, when you try to look at all the data together, that you could say, look, here's the pathway, or here's the physiological mechanism by which that occurs. And I mean, it's kind of, a little bit just the danger of the RNA-seq analysis, because they just produce differences no matter what you do. You turn the culture tube upside down, you're going to see a difference in gene expression. We're at least consistent. So early on, there was an uptake of the fibril adhesins. So as the organism approaches the streptococci, they want to attach because that's what they like to be. So they're getting primed for adherence. And then as they accumulate into those little colonies, where their later colonizers are binding to other PG, not to strep, then they don't need these fibril adhesins so much, and they were being down-regulated. Whereas other factors, such as those involved in human uptake, we know PG likes to suck in human from the blood, and that's where it gets its iron from. And the type 9 secretion system, which is how it secretes proteases to degrade proteins to get peptides for growth and also to release iron, they were being upregulated later. So it was consistent with these being more pathogenic, but it wasn't giving us sort of any additional insights into what might be making P. gingivalis especially more pathogenic when it's with. So we did RNA-seq first, and then we went to sort of metabolomic proteomic analysis. Okay, and so here's the slide you tell your graduate student never ever to present in public, because there's just way too much going on, and nobody's gonna pay any attention for the rest of the talk. But I drew this, so you're just gonna have to look at it for a while. So basically, P. gingivalis has, well, all organisms have what's called as one carbon metabolism. One carbon metabolism is essential, is why all organisms have it. It's essential for producing folate, for one, purines, thymidine, and a number of amino acids are all produced through this one carbon metabolism pathway. Okay. As it one of the precursors of the pathway of one carbon metabolism is a compound called para-aminobenzoic acid, or PABA. So PABA is a precursor that feeds first into dihydrofolate and tetrahydrofolate, which then goes into this cycle and produces amino acids and quite happily on its own produces PABA. PABA feeds into one carbon metabolism. The organism is quite happy. Okay. What happens? The major difference that we were seeing when P. gingivalis was in association with Strep rhodoni, and in this case, it's not with the whole cells because it was a metabolome, and that was just going to make it crazy complicated. So in this case, it was with the supernatant, the extracellular supernatant of Strep rhodoni. And what was in the supernatant of Strep rhodoni is PABA, because Strep rhodoni is producing PABA and produces quite a lot of it. PABA diffuses freely in and out of cells. PABA was accumulating in the extracellular milieu, and P. gingivalis was sucking in this PABA. So P. gingivalis was salvaging PABA, shutting off its own pathway to produce PABA and using the exogenous pathway, the exogenous PABA, to feed into one carbon metabolism. So that seemed to be the major difference that was going on metabolically and proteomically when P. gingivalis was in association, at least with the metabolic extracellular products of Strep pyogenes. So then the question was, well, how does that relate to virulence, if at all? But to begin to answer that question, we looked at, we did a phosphoproteome analysis of P. gingivalis with and without Strep pyogenes. And when we did the phosphoproteome, we found a number of quite interesting things. And first of all, there isn't one association between PG and Strep pyogenes. There's at least two. And that was a little bit surprising at first, but when you think about it, it's really not that surprising. I mean, why would there just be one interaction between these organisms that are closely associated with each other in a microenvironment over a long period of time? So there were two interactions. There was a metabolic interaction mediated by PABA, and then there was a physical interaction that is mediated by the two organisms being attached to each other, being physically associated with each other. But they both feed through a tyrosine kinase, a bacterial tyrosine kinase. Obviously, tyrosine kinases are very well defined in eukaryotic signaling systems, less so in bacterial signaling systems, but they are becoming more recognized and more studied. Tyrosine kinase was inhibited by PABA, but was upregulated by physical association. Tyrosine kinase, through a number of different signaling pathways, controls a number of different properties of P. gingivalis that turned out to be associated with virulence. So first of all, and this again was kind of an unexpected finding, the gingipin proteases. So the gingipins are proteases of P. gingivalis that are predominantly the predominant extracellular proteolytic enzyme that degrades host proteins to release peptides for growth. Right? And P. gingivalis is an asaccharidic organism. It requires peptides and amino acids for carbon and nitrogen. Okay? So it doesn't metabolize carbohydrates. So these proteases are essential for growth. And so the major contribution to extracellular proteolytic activity. It turns out gingipins don't get properly processed and secreted if they're not phosphorylated. They're not tyrosine phosphorylated. The tyrosine kinase phosphorylates the gingipins. Gingipins go through the type 9 secretion system, get processed and secreted, and can degrade substrates, host substrates outside the cell. That was a pretty link between PABA and virulence. And we also find there was a link with the fimbrial expression, which we saw transcriptional. So phosphorylation of some of the transcriptional regulators, CDHR and OPRY, if you have to work on P. gingivalis. So they get phosphorylated, and phosphorylation regulates their activity and the subsequent production of the mRNA for the major fimbria, the FIMA, and the shorter fimbria, the MFA. Tyrosine phosphorylation was also essential for production of extracellular polysaccharide. And that's actually a function that's conserved across a number of different species. That was originally discovered in E. coli, that the tyrosine kinase and tyrosine phosphatase are essential for production and secretion of extracellular polysaccharide. These things, extracellular polysaccharide, makes the organism harder to be phagocytosed, fimbria necessary for attachment, gingipins, which degrade host proteins. All these things are virulence related. So this was a connection between PABA and the whole cells of Streptococcus and virulence. Also, interestingly, and I'll come back to this in a second, tyrosine phosphorylation was important for the function of an enzyme, a PAB-C enzyme, which is a PABA synthesis. So we think this is kind of a feedback mechanism that when there's too much PAB-C activity and balance through the one carbon metabolism pathway is disrupted because there's a lot of homeostatic regulation of one carbon metabolism. But you don't want too much, but you don't want too little of it. So when too much PABA accumulates, it inhibits tyrosine kinase and sort of shuts down the enzyme and helps regulate, sort of self-regulate the levels of PABA. This interaction, the PABA interaction, actually suppresses pathogenicity of P. gingivalis. This is a sort of break on pathogenesis. Before the organisms become physically associated, the metabolites of strep pyogenes are suppressing, are calming the influence of PG development. But when the organisms come in contact, there are mechanisms that we don't fully understand yet, and we're working on these. But there are a number of ways that gram-negative membranes can sense stress and deformation, and we think that might be part of the process. But physical association between the streptococci and P. gingivalis upregulates the tyrosine kinase, and information flow gets reversed in relation to PABA, and we get more of these properties that increase virulence, and so the mice are not happy. Again, in part of a kind of regulatory aspect of this, the physical association shuts down the PAB-C gene, PABA synthesis gene in the streptococci, and reduces the amount of PABA that's being produced. Yeah. Well, yeah, I mean, that's a good question. We don't know if there's any chemotactic aspect to it. So PG is motile, so it's not going to be a strict chemotactic thing, but if there's other ways that it kind of senses where it is. So we think that it's mostly just really related to biogeography, because the early colonizers of the tooth surface, be it above the gum line or below the gum line, are predominantly streptococci. P. gingivalis is an anaerobe, it's a little bit of a later colonizer. So it comes along when the oxygen tension has been reduced by the activity of the streptococci and some other organisms. Then it has a chance to colonize. And the organisms just that it's going to come into contact first, most likely, are going to be these streptococci. Yeah. Oh yeah, I mean that's interesting and brings up a whole sort of set of questions related to the overall community. And obviously, we can only study two at a time. There are, I'll show in a second, there are similar interactions with other organisms. Other streptococci will do the same thing. Other streptococci that P. gingivalis will attach to, which is a particularly small subset or medium-sized subset of the streptococci supported. But yeah, that will allow growth of P. gingivalis. How it affects other organisms, whether it increases those as well or decreases those as well, we don't know. But certainly that's one mechanism that PG, once a colonizer, can start to grow a little bit better in comparison to. Right.
What happens if you use a tyrosine kinase inhibitor? Would you prevent the association of the two bacteria because you have a potential disruption of this biology?
Yes.
Would that be something that
Yes. Plenty of tyrosine kinase inhibitors, most of them are to eukaryotic kinases, and the bacterial kinases are very different. They're very different structurally, and they work in conceptually, yes. Yeah, conceptually, yes. And without the tyrosine kinase, so if we have a TNC library of mutants and we put that into a mouse abscess model, a slightly different model, but whatever, with different combinations of organisms. So you get different combinations of essential genes. The one that's in common between all of those conditions is the tyrosine kinase. Absence of the tyrosine kinase, PG does not do very well in a community. We believe, yeah. So targeting that is certainly, or maybe not getting rid of P. gingivalis, but our idea is more, if we can stimulate this part of the pathway, right, the calming influence predominate, then we could keep these communities under control without having to eliminate PG and something else worse coming in potentially, who knows. But if we could engineer the community so the PG and possibly other pathogens as well are feeling feeling chill, relaxed, not going to be virulent, then that might be a way to do it by inhibiting. Well, yeah, I mean that's interesting, and there may be, and there was a paper from Eric Reynolds' group just a few years ago, and it was, I was probably a student there. There wasn't a whole lot of data in there, but they were looking at the interaction between PG and trap and the glycine exchange and the succinate exchange. But they also noticed there were some of the precursors to the one carbon metabolism were also going from treponema to P-gingivalis. So yeah, it potentially could be working with a number of different organisms as well. And they could be making them more pathogenic or less pathogenic. Yeah. Yeah, that's a good question, and that's an aspect we haven't really got into yet. I'm going to show you a little bit. We've been looking at interaction with neutrophils, but no, we haven't looked at osteoclasts yet. But yeah, that's interesting. That's definitely something we want to look at. Okay, so I mean, you guys kind of dissected all this out already with your questions, but in essence, we think PTK1 is a metabolic couple that balances PABA levels with amino acid levels, which are produced by the gingipains and are substrates for one carbon metabolism to maintain balanced flux through one carbon metabolism. Because again, organisms all have very different and important ways of maintaining the right balance through one carbon metabolism. Basically, PTK1 phosphorylates and activates gingipains. Gingipains produce substrate amino acids for one carbon metabolism. The one carbon metabolism products are essential. The kinase activity also upregulates PABC, so more PABA, but as PABA starts to accumulate, and possibly one carbon metabolism is starting to race away and not be under control, the PABA inhibits PTK1. Less PABA being produced, less gingipain activity, less substrate amino acids, and one carbon metabolism is kept in balance. That's our this. You probably want to see some data to support that. That's always the worst part. But so we've been looking at folate production and gingipain activity and the relationship between those. Folate production first. So folate again is an important product of one carbon metabolism. There's actually a really neat microbiological assay for folate. There's a folate auxotroph for the lactobacillus. You grow that in a standard curve of exogenous folate, then you take your supernatant from your organism of interest, you throw that onto the auxotroph and you measure the OD, and you can, quite quickly get an idea of the level of folate. So here's our wild type P-gingivalis, our parental strain, PABC mutant, all right, so no PABA, no precursor to one carbon metabolism, folate goes way on. Our PTK1 mutant, okay, so less activity of PABC, less gingipain activity, less folate. Our gingipain knockout, so our knockout of the three gingipains, arginine specificity and lysine specificity, so less substrates, less folate activity. We take our PABC mutant and add exogenous PABA, okay, so now the inhibition of block by loss of PTK1 isn't effective, and we get folate being produced again. Can't complement our PTK1 mutant of exogenous PABA because our PTK1 mutant isn't producing enough gingipains. And again, we can't complement our gingipain mutant with PABA. Then we look at gingipain production. So is gingipain production really related to tyrosine kinase and PABC activity? Like I said, there's two different gingipains, arginine specific and lysine specific. They can be on the cell surface or they can be extracellular. So all four of those are decreased in our PTK1 mutant. Genetically complement our PTK1 mutant, they're back up again. Now our PABC mutant was kind of interesting. So our PABC mutant, less PABA, less inhibition of PTK1, and we do see more gingipain activity. Okay, what about virulence? What about pathogenicity? Okay, so we went back to our bone loss model, and these are conventional mice. Again, level of bone loss, sham infection, or wild type infection, and again, like I said, this time we get reasonable levels of bone loss here in this particular. PTK1 mutant, so no PTK1, or potentially the same if we inhibited it. Bone loss is pretty much back to sham infection levels. Then interestingly, when we take our wild type, which would normally give us this amount of bulk, and we pre-incubate with PABA, and we give the mice PABA in the drinking water, we give them PABA every way we can think of, that suppresses the virulence of wild type pigeon development. PABA has this calming effect on the virulence of pigeon development. PAB-C mutant. PAB-C mutant was a little bit of a challenge because if you put PAB-C mutant into the mouth of a wild type mouse, the indigenous microbiota, there's a lot of streptococci in there as well. They're producing their own PABA, probably. I mean, we don't know, but most likely they're producing their own PABA. And so we can't use conventional mice. So we looked at bone loss in a germ-free animal environment. So here's our sham infection. Here's our wild type P. gingivalis. And interestingly, in a germ-free situation, P. gingivalis on its own is not a pathogen. P. gingivalis requires other organisms to express its pathogen. This isn't the initial observation of this. This was described by, as Sal mentioned, George Hashish and Gallus a number of years ago. It's on its own, P. gingivalis is not pathogenic. It requires a community, it requires interaction with other organisms. They make it more pathogenic, it makes them more pathogenic. At our PAB-C mutant on no calming influence of PABA, then we start to see bone loss.
The 3.277 is hijacking also.
Yes.
The microorganisms that we have.
Yes.
In that mouth.
Yes. Yes. Yeah. And would probably be more if we had a lot more streptococci.
Absolutely.
Or other, or other organisms. So the mouse mouth, it's difficult to say even, it's difficult to model, it's difficult to do, and it's very hard to get material from that. So we've also been looking at a mouse abscess model. So P. gingivalis on its own, because they're pretty nice or unpleasant, you're looking from our perspective or the mouse's perspective. Abscess, which you can sample the organisms, you can sample blood from these animals, you can sample the abscess material and look at immune responses. And our PTK1 mutant really doesn't produce an abscess either in this mouse model. We've been looking a little bit about what's going on here in relation to host response, and particularly, as I was saying, with neutrophils. So this is some image analysis where we just inject a luminescent dye that lights up the neutrophils. So we look at neutrophil mobilization, sham infection, wild type. We see pretty good neutrophil mobilization around the abscess area. PTK1 mutant, not so much. We complement the PTK1 mutant genetically, and we start to see restoration of neutrophil mobilization. And then just looking at percent of neutrophils in the blood from these animals, from the tail vein, we can see more neutrophils in the wild type and complemented PTK1 mutant condition. I think a major action. Well, there's really two major actions of the regulation of virulence factors by PAP. One is just the gingipengs are just degrading everything, and that's what P. gingivalis does. But the other is the interface with the host response. Embryo, for example, can recruit neutrophils. Extracellular polysaccharide makes them more resistant to phagocytosis. So we're interested in further studying how it is that P. gingivalis in a sort of low pathogenic state, such as this, differs from P. gingivalis in a higher pathogenic state. In particular, how that relates to the interface with the host immune system. So that's kind of where we are with that particular part of the project. And this kind of summarizes. I probably don't need to go through it all. Just highlight a couple of different things. And the Fusobacterium nucleatum, which is another common oral organism, does the same as the streptococci in terms of upregulating PTK1. Now you mentioned Treponema denticola. We don't know if Treponema denticola does this as well in terms of a physical association. Certainly T. denticola may be impacting the metabolites that are going into 1-carbon metabolism. This PTK1 kinase sort of controls substrates for 1-carbon metabolism, and then controls these virulence factors that can relate either directly to tissue destruction or to the interface with the nutrient. Through distinct states or diminished pathogenicity and an enhanced pathogen. Yeah, it does a little bit. I think for these gingival communities, the polysaccharide, the extracellular polysaccharide, it is not as overwhelmingly important as it is like with a strep mutans infection on the crown of the tooth, where it really requires that polysaccharide to be retentive on the tooth surface. It's not as important as it is classically for some of the Pseudomonas biofilms, where you see these, these textbook structures that are. But it is important, but we don't know what role it plays architecturally in the development of those communities. Okay, so that was how these bacteria communicate with each other directly. Okay, so direct communication, physical and chemical, between P. gingivalis and Strep rhodoni. So kind of like this, they're talking on their cell phone. How did we ever do anything without our cell phones? I mean, when I visited Sal in Boston, we didn't have cell phones. We still managed to meet and have dinner. Last night we texted each other, well, how many times? There wasn't, didn't always have cell phones, right? Back in the day, you picked up your phone and you got through to an operator and you say, hello, this is Mrs. Smith. Can you put me through to Mr. Smith? And the operator would call Mr. Smith and say, Mrs. Smith is on the line. She wants to talk to you. And Mr. Smith like, oh my God, no, tell her I'm busy. We wonder, can bacteria communicate with each other indirectly through the epithelial cells? Okay, because what we'd be kind of looking at is this kind of entity, right? This kind of biofilm entity. It's a cohesive sort of self-contained entity, but also the organisms interface with the epithelial cells that line the gingival crevice, that line the inside of the gum. What we've been looking at is this, but we wondered, can this happen? Okay, can streptococci influence gingival epithelial cells, which then influences P. gingivalis? And one of the reasons we kind of started to think about that, one of the first reasons, we do see microbial communities inside these epithelial cells. This is some work from Joel Rudney in Minnesota from a number of years ago. Again, just doing some fluorescent DNA probes for different organisms. And these are buccal epithelial cells. But you can see there's large numbers of bacteria inside these epithelial cells. This is in vitro work with primary cultures of gingival epithelial cells that we did in our lab, just interacting with P. gingivalis and seeing P. gingivalis can get inside these epithelial cells in pretty. And another part of this is that the P. gingivalis interface with gingival epithelial cells potentially, at least based on in vitro experiments, contributes to a pro-tumorigenic microenvironment. And the association between the oral microbiome and things like oral squamous cell carcinomas is something that has really been increasing of late. And there's a lot of work, a lot of work in this area, a lot of publications in this area. And certainly, gingivalis can do a lot of things. I'm not going to go through them all, but a lot of things that would contribute to carcinogenesis or an inflammatory microenvironment, consistent with tumor development. But we all have P. gingivalis, and we don't all get oral squamous cell carcinoma. So there must be more to it than this. It can't be just P. gingivalis, creates this anti-apoptotic proliferative environment that gives you cancer. So there has to be more going on. So we wondered if other organisms would mitigate the impact of P. gingivalis. It has a lot of its surface proteins and a lot of its structure, like LPS and again the gingival epithelial, the gingival proteases, will do things again. They're consistent with pro-tumorigenic events. But that can't always be happening. Otherwise, we'd all be getting oral squamous cell carcinomas. Again, we started with an RNA-seq. We did an RNA-seq of gingival epithelial cells challenged with P. gingivalis on its own, Strep rhodoni on its own, P. gingivalis and Strep rhodoni together. And what we found here was, interestingly, this is the epithelial cell transcriptome. So this is PG and SG together, very, very different from PG on its own, very different from no infection. And actually, by the first component, closer to the strep cordoni situation. So it appeared that strep cordoni could potentially be reversing or mitigating or suppressing the effect of P. gingivalis on these epithelials. And you see the same things with just plotting the data different ways. PG plus strep versus PG in a volcano plot. You can see they're very different. String analysis. A lot of these nodes that were being differentially regulated by strep cordoni were relating to things that were involved with epithelial cell proliferation and migration. And in particular, we focused on a set of genes that were related to epithelial mesenchymal transition, because most, not all, but most of those genes, when we looked at the two situations with strep cordoni and PG compared to SG alone, the combined situation was much more similar to strep cordoni than it was to P. gingivalis on its own. Not all of them, but a large percentage of them, the PG-SG condition was similar to the SG only condition and not the PG only condition. Okay, so I expect most of you probably know epithelial mesenchymal events better than I do, but epithelial mesenchymal transition is a multi-step morphogenic process by which cells lose cell-cell junctions of polarity and again the ability to migrate. So it's very important. It's a very important normal process. You can do in gastrulation, but if it gets out of control, it's one of the hallmarks of cancer. And being a very important process, it's under very strict control, and it's under control by a number of different transcription factors. And in particular, we focus on the ZEB2 because that was one of the genes that was very strongly differentially regulated by PG and suppressed by strep cordoni. Looking at ZEB2, so ZEB2 RNA again increases by P. gingivalis in a time and dose dependent manner. Being a transcription factor, it doesn't really matter if there's more of it, it has to be in the nucleus. So this is just ZEB2 in green, showing that with P. gingivalis, the ZEB2 starts to localize in the nucleus. And what happens when we add strep cordoni? Well, strep cordoni was able to antagonize the induction of ZEB2 by qPCR being induced by. That was specific to streptococci. Again, we haven't tested every organism there ever was, but most of the streptococci will inhibit PG, whereas things like Fusobacterium nucleatum does not inhibit P. gingivalis. So there is. ZEB2 itself, as an important transcription factor, is under control by a number of post-translational modifications. So we're interested in figuring out, what is it that PG is doing to ZEB2 that strep cordoni is stopping. All right, so we kind of, did our Google image search and we saw the ways that ZEB2 is regulated. So we started working our way through them all. And it turns out, unfortunately, we went this direction because if we'd gone this direction, we wouldn't have wasted quite so much time. Because it turns out it's FOXL1 that controls ZEB2 responses to P. gingivalis. Okay, so if we knocked on FOXL1 with siRNA, P. gingivalis no longer induces ZEB2. And again, this is specific. If we knocked on FOXL3, we don't see the same thing. Of this. So yeah, so the short answer is we don't, and the reason for that is that it's really not a full EMT. So back in the day, it was thought EMT was a sort of binary switch that were either epithelial or mesenchymal, but it really isn't. It's there's a transition, and we think P. gingivalis only gets them a certain way along that transition, not the full way that we'll see morphological changes. Right, so we don't with PG alone, we don't really see a morphotype of the cells. Yeah, that could be part of it, or just that PG on its own, it doesn't just do enough to push it all the way along. It just starts the events, and then it needs something else to complete it. Yeah, that's. We do a little bit. That's, yeah, just kind of anecdotally, the people in the lab say that. We haven't really studied that, systematically, but yeah. Right. Yeah, right. Yeah, dysregulation of apoptosis. Yeah. We definitely see that. Some of these other things, we either haven't looked at or we don't see. But yeah, I mean, we're not quite at the point where we're saying PGN on its own we'll call it a tumor, but it just stimulates some events that are consistent with that kind of environment. So basically, just in a couple minutes, I'll summarize some of this. FOXO itself is controlled by a number of different post-translational modifications. One of the most important, which is phosphorylation on a couple of serine residues, serine 329 and 256. And that determines whether or not FOXO remains in the cytoplasm or goes into the nucleus. So P. gingivalis will dephosphorylate FOXO1 on these two residues, with different kinetics and to different amounts. So PG will dephosphorylate FOXO1 on 329 and 256. But we looked at some, don't need to get into it, did some phosphomimetics to show that those residues are important for ZEB regulation. And they're important for, all right, well, let me describe this a little bit then. We then looked to see if streptogoni would antagonize nuclear localization of FOXO, which is produced by P. gingivalis. So P. gingivalis, yeah, so this is what this is. So green is the FOXO, and so FOXO is controlling ZEB in the nucleus, so no infection. When we have P. gingivalis, we get a lot of FOXO going into the nucleus. Streptogoni doesn't do that, and when the two together, FOXO tends to stay in the cytoplasm. That's something we haven't done yet. Then we find that so this dephosphorylation of serine 329 seems to be the most important. That streptogoni, in addition to antagonizing nuclear translocation of FOXO induced by PG and ZEB regulation, also activated this NEMO-like kinase, this NLK pathway, which is phosphorylated again to some of threonine 298, stimulated by streptogoni, even in the presence of P. gingivalis. If we knock down NLK, streptogoni loses the ability to antagonize induction of FOXO1 by P. gingivalis. This is as simple as readout with a FOXO reporter. NLK, NEMO-like kinase itself is controlled by another kinase, TAK1. And same kind of thing, when we knock down TAK1, we see streptogoni is no longer able to antagonize the ability of P. gingivalis to activate FOXO1 and to induce translocation of FOXO1 into the nucleus. This is our control, FOXO1 in the nucleus with PG, antagonized by streptogoni. We knock down NLK, or we knock down TAK1, and streptogoni is no longer antagonizing P. gingivalis. Yeah, I think it probably is a little bit just because this pathway, this TAK-NLK pathway, does activate FOXO1. Okay, so basically, if we look at this side of the figure first, so P. gingivalis, and I've drawn it intracellularly here, but we don't strictly know that it has to be intracellular to do that. Okay. P. gingivalis dephosphorylates FOXO1, FOXO1 goes into the nucleus, FOXO1 controls that. And then these are the epithelial markers and the mesenchymal markers and the stemness markers that we've looked at, which are differentially regulated. So we've done these, but we haven't done anything more than that. And morphologically, we don't really see it. Streptogoni on its own Streptococci stimulates TAK1 phosphorylation and NLK1. NLK1 prevents PGN-Gibbs from dephosphorylating FOXO1 and prevents this association. This PGN-Gibbs, nothing's ever that simple, and PGN-Gibbs also has different ways that it can activate ZEB1. And it can do that by activating PI3K, AKT, going through GSK3 and beta-catenin, and beta-catenin can control ZEB2. So in addition to having this dramatic effect on ZEB2, we believe PG has a pathway that's insulated from streptococci that can continue to regulate ZEB2 to some degree, obviously not to the degree that's causing this major epithelial mesenchymal shift, but we think this interaction or this regulation of ZEB2 is important for intracellular survival of the organism. So there's more than one way to accomplish that. And honey inhibits one of the major ways and sort of shuts down that potentially pro-tumorigenic event. But PG is still able to manipulate ZEB2 to some extent. So I'll just summarize. PG and escrodoni develop in the communities, which are synergistic, and pathogenicity increases over time. PABA regulates expression of PGN-Gibbs fimbriae through tyrosine phosphorylation, regulates gingipan activity through the 1-carbon metabolism cycle pathway. Epithelial cells, PGN-Gibbs transcriptional profile is reversed towards homeostasis by streptococci. Inhibits this FOXO-ZEB2 axis by antagonizing dephosphorylation of CM329. The protective effect of escrodoni is mediated through its interactions with host epithelial cells rather than direct bacterial interactions. So the role of streptococci, just going back to this context, all life exists in context kind of thing, the role of streptococci in all communities just depends on the situation. In periodontally relevant communities, subgingivally, it's probably an accessory pathogen. It makes PGN-Gibbs more pathogenic. For an epithelial cell surface, it does the opposite. An epithelial cell surface working through the epithelial cells that has a protective role. And in that context, we like to think of it more as a homeostatic commensal or a health-associated commensal. Thanks to all the people that have been working on that. These are the current members of the lab. Obviously, there's been a lot of people over the years working on these projects. Our collaborators at the University of Louisville, our collaborators at other institutions, NITCR for funding, NIGMS that funded our germ-free animal facility. And, when you're from Louisville, you're legally required to show a picture of bourbon and horse racing. So if you ever come visit Louisville, I can certainly show you plenty of bourbon. Depends on the time of year for the horses. Okay, so thank you very much for your attention. Yeah. Yeah, that's an interesting question, and short answer is I don't know. I mean that's interesting. We may do. There could be metabolic effects based on the gingipans, but the gingipans are really kind of toxic to the host tissues. They degrade structural components. They degrade antibodies. They degrade cytokines. So it's hard to see what would be the beneficial effect, but low levels, maybe. Yeah, maybe it's certainly a possibility. Yeah. Really in humans, probably not. We all have a large number of streptococci. Newly erupted tooth within seconds is covered in streptococci. You go to the dentist, go get your teeth professionally cleaned, you get all that scraped off. Oh, that really feels good. The streptococci are back on there. Yeah, you wouldn't, I mean, you wouldn't get rid of all of them, I don't think. You suppress them. Yeah, you suppress them. Yeah.
Following up on that one, so if someone is on a long-term sulfur drug treatment, so is that going to give an upper hand to
Yes, potentially, yeah, yeah. I mean, theoretically, yeah. Not something we've really been able to study very well in the lab. I mean, we looked at that because that was one of the early things we thought of. Oh, yeah, well, that's what the sulfur drugs do. A lot of other organisms, you disrupt one carb metabolism, they're less pathogenic. You know, probably part of how those sulfur drugs work. Whereas in PG, it's the opposite. They're more pathogenic, possibly because of the asaccharidic lifestyle. Yeah, they may get a bit of an advantage. You know. Yeah, we did initially think we could look at just the transcriptional level of some of these genes and back from healthy people and diseased people, and maybe get an idea about that, but never really went very far, possibly because a lot of the regulations post-translation. So it's going to be a little bit harder to measure kinase activity. And then just the variability of collecting samples, you know, where you get them from. Does a person really have disease? Is active, or did they have disease 20 years ago? Kind of thing. So yeah, we'd love to be able to do that, but we just, you know, we need a sort of clinical tie-in that is more than we have at the moment. Yeah. Oh, probably not. Just, you know, once a day swishing and spitting probably isn't going to do anything. Alcohol is more people who are consuming, yeah, large amounts for long periods of time, and are, you know, are kind of reasons. No. It's, you know, a shot of bourbon a day is absolutely fine. Yeah, thank you.